Our next speaker is Dr. Anita Oberholster. She runs a great wine lab. Uh, we work a lot together, so I'll turn it over to her. So.
Thanks, Vaughn. Good afternoon. It's my job to make you all stay awake, right? I did say wake up. I said stay awake. Hi, everyone that's online. Okay, so we have a few minutes. So if you're sitting next, anybody at the back that would like to taste wine, move forward so that we can pour wine for you. Otherwise, if you have a glass next to you and there's nobody sitting there, do you mind just putting it upside down so the pourers know not to pour it? We're going to quickly pour the wine because we have 5 extra minutes. I apologize for those that's online. You have a very quick break. Come back in time. Only 5 minutes. We're going to quickly pour the wines. I gotta get you. Okay. I'm back. I just stole water. This is a weird pointer. It doesn't go where you point it. I'm entertaining the people that's online. So pourers, remember to pour for yourselves as well. Sorry, they're mostly people in the lab or students. This is a good educational moment. Anybody missing a wine, then just raise your hand. So please don't get so absorbed in my prize-winning wines that you forget to listen to my presentation. Just saying. Those are experimental wines, so don't have too high expectations, is all I'm saying. Also, try not to, we're going to taste them at the end. See in the first 2 glasses, I don't want to tell you what they are. This is why you get A, B, C, D, E, F. But see if you can save a little bit of wine in the first 2 glasses and then a little bit of wine in the last 4 glasses. I want to do a little bit of blending, but you need very, very little to do that. Okay? And it doesn't have to be very exact either. Sorry, we're just giving out crackers at the moment. Good? Okay. Sorry, I apologize for the people waiting online, but it can be very disturbing when people walk in front of you and try and pour wines with somebody else, or you're trying to listen to a presentation. So we had 5 minutes, so I thought, let's use the 5 minutes to pour the wines. So, my talk. So I'm going to talk about the impact of red blotch disease on grape and wine composition and potential mitigation strategies. And I'm also going to give you a recap, a very quick recap of the work that we've done since 2014, because that's when I started working on red blotch disease. So I'm not going to give you any background because I assumed as I was last that everybody would have told you all about the virus and about its potential vectors. So initially, basically, what we set out to do was, it was a newly identified virus, and we really didn't know the impact, except for anecdotal evidence. We didn't know the impact on grape and wine composition and how that would relate to a wine quality impact. So the first year, what we did, we basically tried to work on a few varieties, few different sites, multi-seasonal, to see what is the impact. So we had different sites in different regions of Chardonnay, Merlot, and Cabernet Sauvignon. And basically what we did on the grapes, we only harvested them at full ripeness, at 25 brix for research purposes. And we harvested the red blotch positive. So this is those that tested positively for grapevine red blotch virus, or in the case of winemaking, we couldn't only use tested vines in the beginning, those that showed clear symptomology. And I have to say we had 100% correlation at the sites we worked at between testing and symptomology, because these were vineyards that have been monitored for a long time, so we could trust the symptoms. So basically, in the grapes, we looked at mostly phenolics as far as secondary metabolites go, because at that stage, everybody knew they could see an impact on color development, and we looked at primary metabolites because everybody could see an impact on sugar development and sugar accumulation. So this is our main findings in those 2 years looking at these different sites. We saw that both primary and secondary metabolites were significantly impacted. We saw consistently a decrease in sugar accumulation. You can see the range there for Chardonnay, Merlot, and Cabernet Sauvignon. So it's quite a big range. And you can see 1 year there was almost no impact on the Chardonnay. We saw an increase in amino acids and organic acids in the red blotch positive fruit. And this is a side effect of decreases in ripening, because at the end, some amino acids and a lot of organic acids do decrease when you get to that ripe to overripe stage. And then we sometimes, not consistently, saw this increase in skin tannin in red blotch positive grapes. Now, it wasn't always significant, and there can be two reasons for that. Many times, the vines that showed symptoms of red blotch disease would have less vigor because you have the leaves turning red and shriveling up. So you can have higher or more sun exposure that can increase skin tannin a little bit. Or this could be a defense mechanism of the plant because the synthesis of tannin is part of the defense mechanism of the plant. And we still don't know for sure which one is the driving force here. Then we saw definitely consistently in red grape varieties a decrease in biosynthesis of anthocyanins. And we did some transcriptomic work, and the lab of Professor Cantu also did some work and confirmed that grapevine red blotch virus can inhibit or delay the ripening processes and the biosynthesis of compounds in the phenylpropanoid pathways. And that's the pathway that synthesizes anthocyanins as well. So we have seen this. And that's the most common impact. The tannin impact was a little bit inconsistent. And we saw a lot of variability depending on the site and the season. Then we also made wines from these grapes that we analyzed. And in the wines, we did the same thing. We looked at primary and secondary metabolites, focusing mostly on the non-volatile, the phenolics. And then we also did descriptive analysis because that's the best way for us to see, okay, we see differences in analysis, but does it actually relate to a sensory impact, a quality impact in the final wines? So what we mostly saw was that, yes, there were differences, mostly alcohol differences due to the large sugar differences you can see if you harvest red blotch positive and negative, so healthy fruit and grapevine red blotch virus infected fruit at the same time. Then we also most consistently saw a phenolic content difference that could actually relate into sensory differences. So if you look here, this is for the Chardonnay site, the first year. If you look at the sensory, this is a principal component analysis. The score plot of our wine treatments based on all the attributes, 18 attributes that we rated during the sensory evaluation. And you'll see you can clearly differentiate between the negative and the positive, but only one attribute was significantly different. Okay, so many times what you see here is a synergistic impact of small differences, but it doesn't really relate to large significant difference in specific attributes. So it was not a major impact. Then we did this for another year. And in 2015, this was the difference. A significant increase in apple juice character in the red blotch positive, a significant decrease in hot mouthfeel. This is due to alcohol differences in the red blotch positive wines. Now, this specific producer, after seeing these results, decided not to pull the vines that were impacted, but rather to wait until that specific vineyard block would come in for replant in any case, because the impact was so small on the Chardonnay. However, there were yield impacts. So for all these sites we looked at, the yield impact wasn't consistent. In some years, it was barely there. In this site, it was consistently around 15% decrease in yield in the vines that were impacted by red blotch. If we look at the reds, so this is now 2 Cabernet Sauvignon sites and 1 Merlot site. And you'll see the impact of site was larger than the impact of red blotch disease. However, if you go into each site, the story does change. Now, I'm going to only show you one as an example. So this is the one where we saw the largest differences. And this is at Oakville Experimental Station. And what you see here is a clear difference between negative and positive, and this was due to 3 significantly different attributes. A significant decrease in astringency, a decrease in hot alcohol, that's on the nose, and decrease in alcohol in the mouth, right? And this related to phenolic differences, significant decreases in anthocyanins, polymeric pigments, polymeric phenols, and percentage alcohol in the red blotch positive wines compared to the wines made from healthy fruit. And really, if you tasted these two, the difference really doesn't come through if you only look at significant differences of a specific attributes. It was very, very, very clear, and it had a significant quality impact. So one of the sites we worked with asked me an interesting question. They said, okay, so when do we need to start being concerned of having red blotch in our vineyard? And they had a very ideal situation where they had red blotch due to planting. So they had 2 rows that wasn't impacted and 2 rows that were 100% impacted. Planted at the same time, everything identical, no spread for some reason. And so what we did is we harvested the positive and negative at the same time, but separately. And then in the vineyard, in the winery, combined certain percentages of red blotch impacted fruit. So here, that's 100% healthy fruit, included 5, 15, 25, and 100% of the red blotch fruit. And this is what we found. Very interesting. So at the top, it's a little bit small. So just believe me, if you look here, that is 25% and that's 100% red blotch impacted fruit. It's here on the left. You can see way more aroma compounds on the right. So we started also looking at the volatile compounds because when we started doing the sensory analysis, it became clear that the impact wasn't only on the mouthfeel, on the phenolics, there was also an impact on the volatiles. Now, the left, what you have here is some compounds that really gives you more green floral characters, and the right is more your fruity, jammy characters. If you look here in the descriptive analysis, you can see these were the attributes that were significantly different. Effervescent was just because of some gas, so ignore that. What you want to look at is sweet, hot, bitter. Now, something to remember, alcohol. Alcohol decreases your perception of astringency, increase your perception of bitterness. So that would be part of the reason, could be part of the reason why there's increased bitterness. And the hot and sweetness is also due to an alcohol perception because these wines were all dry at the same level. So, and what you can see here clearly is for this case, as soon as you had more than 15% of red blotch impacted fruit included in your fermentation, there was a clear difference that the sensory panel could pick up. And if you look at the analytics, if you look at anthocyanins, you can see as soon as you got to more than 15% inclusion of red blotch impacted fruit, you had significantly lower amounts of anthocyanins in your wine as well. But now, I'm not saying this can translate to any vineyard, right? In this specific year, the impact due to red blotch was 3.6 brix difference at the time of harvest. Now, we harvested when the negative, the healthy fruit, was at 25 brix. So basically what we said, in this specific vineyard with that kind of impact, if you had more than 15% incidence of red blotch in your vineyard, they really should have done sequential harvesting and kept the red blotch fruit separate, because if they included it, there was a clear quality impact, and this was on the Napa Valley floor of this vineyard, so I can tell you it goes into a very expensive bottle of wine. So that was important information for them to know, and they immediately pulled all the red blotch wines, so I couldn't study this site any further. But this keeps on happening to me. But the thing here to remember is I would suggest if you have red blotch in your vineyard, make the effort of taking your ripening samples separate. So you can see for that specific season, what is the impact? Because seriously, there's a huge seasonal impact. So if you know the impact is significant, and yes, when you get to like a 2 brix difference, and you have more than 15% incident of red blotch, you may want to consider doing sequential harvesting, may want to consider making those wines separate, and see what the impact is, and whether you can do the blending rather after the fact than being stuck with it in your main fermentation. Something that we're working on that I'm not going to talk about today very much is we have been doing some transcriptomic work, and we have started to get an inkling of why there's such a huge seasonal impact. Now, it seems we had 1 year where we had a heat wave just after variaison, and it seems the virus titer, and we're still investigating this, is very high at the point of variaison. And with higher temperatures, we saw a higher accumulation of short interfering RNAs. Basically, this is which is responsible for virus gene silencing in a vine. It's part of the response of the vine to the virus to protect it against the virus. And you get more of this silencing happening at higher temperatures. And so many times, if you have high temperatures at certain key stages, you see less symptomology of grapevine red blotch disease, which is very interesting. But we've only observed this in one season, so we are investigating this further. But this is part of the reason you see such a great seasonal impact, is due to mechanisms like this that has an influence. So then we realized, okay, so perhaps we should not be harvesting the negatives and the positive at the same time, right? Because alcohol has such an overpowering impact on all our sensory evaluations. So we started doing sequential harvesting. We will go in, we will harvest the positive and the negatives on the same time, and then go back aiming to get those positive vines also at 25 Brix, so that we can compare them at the same sugar levels and only look at the impact of the virus on everything else other than sugar and alcohol. So we did this at 2 different sites. We have a site in Paso Robles with Merlot, and an Oakville site that actually was part of a rootstock trial many moons ago. So we have 110R and 420A. 110R is slightly more vigorous than 420A. And this is in 2016, this is what we found. So you can see the variable impact. So this is a 12% decrease in sugar specifically. And even here, this is interspaced vines. They're in the same block. And you can see for 110R, there was a 15% decrease. And for 420A, 9% decrease in sugar accumulation. If you compare the positive and the negative at the same point of time when we harvested the negatives at 25 Brix. Then we tried to go back and get to 25 Brix a little bit later. We weren't always very successful in that, as you can see. We try. But at least the differences were a little bit less. So this graph Kant showed you, this year we worked together and they did a lot of the grapevine physiology measurements for us. And something I want to point out here, and this is important, is that longer hang time does not give you more anthocyanins, right? Many times when you start getting to that ripe to overripe stage, even if your sugar levels are low, you do start getting degradation of your cell walls and many other things. We saw a decrease in anthocyanins in both your negatives, your healthy fruit, and in your red blotch impacted fruit. The same with tannins, everything else. We didn't see an increase. You saw an increase in sugar only due to dehydration. But we didn't even see an increase in concentration of our phenolics in the grapes because they were actually, it seems to be either less extractable or degrading, okay, in the grapes itself. So what we're doing here is we look at extraction during fermentation. So remember here, basically, there was no more phenolics in the fruit that were harvested second, with the second harvest, okay? The sequential harvest red blotch fruit. So if you look at the green line, that is your healthy. The solid red is the second harvest red blotch impacted fruit, and the dotted lines are the red blotch disease fruit that was harvested at the same time as the healthy. And you can see consistently, no matter the site, you saw that second harvest actually helped. And I'm just telling you, there wasn't any more anthocyanins in that fruit. We saw the same thing for tannins. Basically, there can be 2 potential reasons. One is higher alcohol. The second harvest did have slightly more alcohol, or more sugar, and resulting in more alcohol. And alcohol, we know, can result in higher solubility of phenolics. It can result in less reabsorption of phenolics onto cell wall material, onto yeast and other solids in your fermenter. Or it can be due to higher extractability. Cell wall degradation, right, that takes place during ripening, more extractability from the cell walls itself. Then if you look here, this is the molar results. And it's quite interesting. If you look here at the PCA, you'll see once again, only the significant attributes are shown here. And most of this is related to alcohol, right? More bitterness, higher alcohol on the mouth, on the nose, etc. Here's color perception. So they actually evaluated the color. And here you can see there really wasn't a clear significant difference between the negatives and the second harvest positive fruit. But those that were harvested at the same time after the negatives, you can see there was a significant decrease in color. And we know color is a very important factor when we think of quality for red wine. Now this is for the Cabernet Sauvignon. This is 110R rootstock and this is 420A rootstock. So green, healthy. Red, red blotch disease. Yellow, second harvest, red blotch disease. And what you can see here is they did move closer. They could still be distinguished from each other, but they did move closer to each other. And your red blotch positive fruit harvested basically pretty green, at 22, 23 breaks. They were correlated with unripe fruit aromas and sour taste. So second harvest did improve things because of higher extractability of phenolics. The volatile seems better. It had a little bit of better alcohol, closer to that of the negatives. So it did seem to have a positive impact, although you could still distinguish that red blotch fruit from the negative. So in the next year, we went, okay, let's add a treatment. So now we had the negatives. We have the positives. So NS means no sugar added. Then we added sugar to those red blotch positive fruit that we harvested at the same time as the negative. So we wanted to remove the whole difference due to alcohol content. And then we did try to do second harvest, also aiming for 25 bricks of the red blotch positive fruit, so it has the same sugar content as that, right? So if extractability differences were due to alcohol differences, then if we hit the mark, these two should be very similar, right? Okay. So this is what we found. So green, healthy. Red, second harvest. Oh, sorry, I'm going to confuse everybody here. Green, healthy. Orange, yellow, whatever you see there, the lighter one, that is second harvest, red blotch positive. And then the red is red blotch positive, harvested at the same time as the negative. And the blue is the red blotch positive, harvested at the same time as the negative, with sugar added in the fermenter. Okay, make sense? And you can see the sugar addition didn't seem to do anything. And this was true no matter what phenolic we looked at and no matter what site we looked at. There was a consistent impact. So this increase in extractability was not due to the higher alcohol. It was due to what we presume would be cell wall extractability, right? An impact due to the cell walls and the longer hang time. So this is a multi-factor analysis. So this is a little bit different to just a principal component analysis, because here we have everything we measured. We have phenolics, basic chemical parameters like alcohol, TA, etc., pH. We have sensory, and we have the volatiles that we analyzed. And basically, it can show you, if you look at all these, that basically for 2016, you have your second harvest is there, this is your healthy, and this is your red blotch positive. So they're all separated from each other. Now, the second harvest is closer to this one sensorially and phenolically, but, oh no, not by phenolics, but really sensorially. But they're still separate, but they're more similar. If you look at 420A, similarly, they're different, although you can see why they are different and what is the most similar. So here, because of their phenolics that are more similar to the healthy, these two are actually more related and correlated to each other, but they're still different, okay? If you look at 2017, so here we have an extra treatment, right? Here we have the sugar chaptalization, positive, second harvest positive, and negative. Now, the interesting thing to note here is that the chaptalized treatments, and it's not that clear to see here, but the chaptalized treatments were closer to the negatives due to the volatile analysis being more similar. So the volatiles were actually seen as more similar when we did chemical analysis. But sensorially, that didn't clearly come through because obviously alcohol has an impact. It can increase volatility, but when you're sensorily trying to perceive something, alcohol can also be an impediment. But it's very interesting that volatile-wise, chaptalization did work. But it did not help for phenolic extractability, and it did not help for the mouthfeel differences among these wines. Other than helping the alcohol, it didn't help for phenolics. So that was very interesting. So it moved closer, but it was not a 100% answer. Second, longer hang time had a better impact than chaptalization. Now for 420A this year, you can see we don't have a second harvest, but if you think 2017 Oakville, you will realize what happened. We had smoke. So we made the wines, but we couldn't do sensory because they were smoke impacted. So that would have influenced all our results. So basically, what's the main thing that we noticed then? We said, okay, decrease alcohol content if you have red blotch positive fruit. And that was significant. Decrease in anthocyanins and color in your final wines. That was many times significant. Trend of decreased tannins, but it was not always, right? Sometimes there was no difference, and sometimes the red blotch positive fruit could actually have a little bit more skin tannin. In general, the wine seems more sour, more green aromas, more watery, less body than those made from healthy fruit. But like I say, it depends on the season and the site, that differences aren't always that clear. I've had years that people could not distinguish between the red blotch positive wines and the wines made from negative fruit. That has happened too. So don't assume this is always the case. And I would definitely suggest that if you see a large difference in sugar accumulation between your virus-impacted fruit and your non-impacted fruit, to think about, and you have more than 15% incidence, to think about sequential harvesting before you make the wines. And definitely every year you need to test, because you can't assume because it was like that last year that this year is going to be the same due to the seasonal impacts. So at this point in time, longer hang time has been the best thing that we could do. It seems to be basically you get the increase in primary metabolites, for specifically sugar, due to dehydration and no other reason. You get higher extractability, it seems due to cell wall degradation during ripening. And that is something we're going to now investigate further, because we want to know why is this happening. So if you think about cell walls, during the ripening, in the ripening grapes, you have beta-glucan enzyme degradation, and that would many times increase anthocyanin extraction. And we know grapes with higher anthocyanins and higher skin tannins, and if that results in wines with more skin tannin and more anthocyanins, that is the one thing that we know correlate with higher rated wines, higher rated red wines specifically. So we thought, okay, let's think about this. First thing we need to investigate, what impact does the virus have on cell wall development and cell wall degradation during ripening? But secondly, what can we do potentially in the winery to negate this impact, right? Could we add maceration enzymes? Will that actually make up for the difference in the cell wall? Or can we just do extended maceration, and would that make a difference for the impact we see due to the virus? So we did this experiment, and because we also see the longer hang time issue, we thought, okay, commercial harvest, in fact, we all know is closer to 27 brix than 25 brix. So we will do both, right? So we went 25 brix, positive and negative, and for the positive, EZ is enzyme treatment in the winery. EM stands for extended maceration. And then we went back for 27 brix and did the same thing, where our control negatives are positives. Now, obviously, the dream was that these positive, that 27, would be similar to that 25, right? Because in this specific site, we most consistently saw a 2 brix difference by the time of harvest at 27 brix. Now, obviously, those dreams never work out when you actually do it, the season you're actually doing the experiment. So there's a little bit of a small table, I apologize, but if you look here, you'll see our 25 brix was actually 26 brix, and our negative, and our positive at that same point in time was at 22.8. We went back 2 weeks later, and we got that aim, 27 brix for the negative, and our positive was, guess what? 22.6 brix. It didn't move anywhere. It got stuck, right? Which sometimes happens. Has never happened at this specific site, but this is the year it decided to happen. Good omens. Okay, now this is a little bit of a busy graph, but okay, let's try and not get confused. Okay, if we think about anthocyanins, so this is the graph here on the left. Higher extractability at higher brix. Okay, so if you look here, the dotted lines is the higher brix for the negative grapes. The solid lines is the lower bricks, okay, for the same treatments. Now, red blotch positive, those are the red wines. So you can see even there, the solid line and the negative line, it didn't really make a huge difference. But in general, we saw more extractability at higher bricks. Then, if you look at the enzymatic treatment, it did not increase extractability. If you think the enzymatic treatment is the blue line, the blue lines and the red lines are pretty much on top of each other, correct? Then if you look at the purple lines, that's extended maceration, didn't make any impact. Now, I didn't do extended maceration thinking you're gonna get more anthocyanins because you don't, right? Anthocyanins extract within the first 5 days for a hot fermentation, but we did think perhaps we'll get more tannin and that will help for the thinner body and perhaps have an impact there. So if we look at the tannin here at the bottom, you will see that actually with higher bricks, we got less tannin extracted, which was interesting. Now, there's some research from Australia where they showed that with increased ripening, the porosity of your cell walls increase, and that led to higher reabsorption of tannins, especially higher molecular weight tannins, back onto the skin cell wall. So it could be something like that going on. But other than that, if you look at the enzymes, both the enzymes helped, and both extended maceration helped, and you got more tannins out of the grapes. Okay, so it helped for tannins, didn't help for anthocyanins. So this is the wines. So unfortunately, as you can see there, the negatives pretty high alcohols, the positives pretty low alcohols, so that will have an impact as they got stuck and didn't get ripe. And so this is, if you count up the years, you would realize that this was a COVID year. So this is the sensory results, and we weren't allowed to do the sensory, so I had to outsource it. So this went to a company that has a dedicated panel, and they do RATA. RATA means rate all that applies, right? So we do bench trials, we say what attributes we see in the wines. This is a dedicated panel, and they rate those attributes. So unfortunately, it means that the results had large variability among the different fermentation reps. So I gave them 3 fermentation reps for each treatment, trying to get better data, because we analyze everything, even sensory, in triplicate, which a company is not going to do, or they will do if I had enough money, which I didn't. But still, if you look here on the right, see the yellow? That is the negative 25, okay? So everything else you're seeing is something positive and a treatment, okay? So there was some separation. And if you look on the right, you will see alcohol-related terms, you will see jammy, spicy, body weight, astringency, everything you like in your full-bodied Napa cab, right, kind of on the right. Okay, this is not a cab, it's a Merlot, so that was a mistake. But, you know, everything you would like in a good Merlot, can I put it that way? So what I did is this one will show you a little bit more clearer. So this is a spider diagram, and what I did is I took the means of the treatments that were the most similar based on the sensory. And if you want to summarize it, you can look here at the left. I tried to compare the two for you. So the positive was greener and had less body. This is something we see quite often when there is a significant impact. The longer hang-down positive was more similar to the negative than the one that was harvested at the same time. Okay, so this supports what we've now been seeing for several years. The enzyme treatment and the extended maceration did increase the body of the wine slightly because of that extra tannins, but it also increased the bitterness. So that was, for me, a negative. And also, because these grapes was still green, right? There were 22, 23 bracts. That extended maceration actually, for me, enhanced like a green stemmy character in the wine that I'm definitely not very partial to, and I think most other people aren't. So for me personally, it did not increase the quality of the wine. So we end up still saying longer hang time still seems to be the best thing we can think of, right? Trying to get that red blotch positive fruit ripe, if you can. And even if it doesn't move in sugar value, just that degradation of the cell wall seems to make a difference and give you a higher extractability that helps. Any kind of chaptalization, concentrate, whatever, will help the volatiles, but it's not going to help for anything else. And it will help you with your alcohol difference as well. If you see a large difference in your vineyard, do sequential harvesting so you can blend afterwards. And that gives you a little bit more control over the impact of this virus on your wine quality. And I do think that maceration enzymes, we've only done this once. We have seen studies where maceration enzymes were able to increase the extractability of anthocyanins. We didn't see it this time. So, it may be worth still playing around with this in combination with longer hang time, and perhaps someone else may see a better impact on a different site. So what's next? We want to identify the soluble proteins and pectins in cell walls. So we've done some preliminary work. We've seen there's an increase in grapevine red blotch virus infected grapes in their cell walls have higher proteins. This could be pathologist related, so this is basically a response to the virus. And we know proteins in cell walls could potentially retain tannins so that you can get less extractability. We want to investigate the impact of virus titer on symptomology. Is there any kind of relationship between the two? We want to look at the relationship between years of infection and symptoms. So we have a very nice site where we now have 4 years that we know exactly when the vine got infected, or I should rather say, we know when it got infected, we know when it started showing symptoms, and we're actually making separate wines from these vines to see, okay, is there an impact? Can we see a direct correlation? And we're doing it now for several seasons, so that will be very interesting. And all of this is at the end to determine what is the economic impact to help people to make decisions. Because at this point in time, the best solution for grapevine red blotch virus is still roguing, right? And that has a huge economic impact. When do you hit that tipping point that is the best decision for you to make? So that was a long story. This is some of the people in my lab and some of the funding we've gotten through the years. We're very, very thankful for funding. JLR Vineyards got me started, my first experiment, and now currently we're all part of a large USDA grant that we're very thankful for. So, tasting, the best part of the day. So, I'm not going to tell you what you're tasting, but you have a good idea because I just discussed the wines. But so, if you want to make a few notes for yourself, in front of yourself, and try and save a little bit of wine in glass 1 and 2, A and B, and then save a little bit of wine in, it doesn't really matter in the last 4 glasses, but say wine 3. Okay. And just, I'm going to give you a few minutes to taste. I'm going to retaste them, and then we're going to discuss. You are all poker players. I can't see anything on your faces. Do I have any online questions? I assume not. No? I'm going to assume it's not because everybody left drinking coffee, having somewhere it's late enough to have a drink. But because I was so clear. See, the wines are so lovely, even though you just got a 1 ounce pour, everybody has wine left in their glasses. But it's good. You want to go back to it when you actually know what you're tasting. Okay, are we ready? I wonder how many people have been able to work this out. Okay. So, the first two wines is the negative and the positive, harvested at the same time. When it says 25, because that was my treatment name, but we know that a negative was actually at 26 bricks, okay? Could anybody see a difference between those two wines? It was pretty obvious, right? I mean, for me, the positive is definitely thinner, more sour, more bitter, more muted fruit compared to the negative. Would you agree? Yes? No? Don't know what you're talking about, Anita? Okay. Now, my experiment. So remember when I was talking about mixing 100% impacted fruit with non-impacted fruit and the impact that it had? Now, I actually played around with this because I like doing stuff like this in the winery. And then I had a measuring cylinder, but you don't have measuring cylinders, so we're going to see how good you are at baking. So here's your negative, here's your positive. If you think of the two, just pour a drop. So I don't know, what would be 15%? I have no idea. Whatever you think is 15%. Just pour the positive into the negative, and then you taste it again, and then you tell me if you can actually notice a difference. Somewhere between 15 and 30%, that was it. So for me, don't want to influence anybody. What I noticed when I did it was that the fruit gets muted for me a little bit. If you put the positive into the negative, and you immediately get more of that green character by mouth. And it's more like a stemmy green. I'm not talking red, a bell pepper or stuff like that. I'm just talking more like a sour stemminess by palate. That's what I personally notice. Well, nobody's screaming and telling me how wrong I am, so I'm just going to go with the flow here. Okay, now, for what do you think about the Red Blotch positive at 27 bricks compared to the 25 bricks? I mean, obviously, it wasn't actually 27 and 25 bricks. They were both at 23 bricks, but the one was 2 weeks later and had a little bit higher extractability of phenolics. Did you notice a difference between those wines? They have a little bit more, I mean, it's not like the fruit suddenly pops out and hits you in the face, but it is a little bit more riper candy, cooked fruit kind of characters than in the one that's harvested at the same time as the negative at 25 bricks, for me personally. And on the palate, it still has that stemmy, but it has like a dried fruit stemmy character. It's just a little bit riper impression overall, but not, you know, a miracle fix, like I'm saying. Now, if you look at the two that was the positive and negative that was harvested at the same time, 2 weeks later, and you do the same thing. You pour a little bit of, say, 15 to 30% of the positive into the negative. What do you think? Does it improve the wine if you include some of the red blotch positive fruit? No? No one? No, it does not. I mean, the impact is for me greater on the 25 side than on the 27 side, but you still get specifically on the palate, you get a more greener, sour, bitter impression than you get from the wine that's negative. So, do anybody think, tasting these wines, that the enzymatic treatment, maceration enzyme, and the extended maceration helped? No, I see a few. See, I don't lie. See, this is the show and tell period. I told you the sensory said it didn't really help. It really didn't help. There's more tannin there. There's slightly more weight on the palate. The negative about that is because you have a wine that is pretty green, a pretty sour impression, that extra tannin, even though it gives you a little bit more weight on the palate, is not actually improving the quality. It actually makes it, especially the extended maceration, gives it that seriously bitter, green, stemmy, it's like, would you do extended maceration on fruit that you know is not optimally ripe and have green seeds and green character? No, you're not, right? So, when we said we're going to do the extended maceration, we didn't actually put a time limit on it. We thought we will taste it as it ferments and then stop it when we thought, okay, this is not moving closer to the negative, this is moving further away from the negative. And after a week, it was just getting more bitter and more green by mouth, so we just stopped it after a week, right? I didn't see any point in extending it because I realized 2, 3, 4 weeks, it's probably not going to get better. So that's why we only did it for a week. Our regional plan was to actually do it for at least 2 weeks or longer. But I do think the one with the macerating enzymes is better than the extended maceration, I would say. It's less, it's not as bitter. But clearly for these wines in this year, and you can see that the impact was basically 3 bricks. 3 to 3.4 bricks at the time of harvest, right? So that is something I see as significant, if that's the impact on the primary metabolites. And you could clearly see the impact on the wines. And this is straightforward, simple wines, right? This is experimental wines. So they're made identically the same, EC 1118, right, yeast, or 1118, however you like to say the name. Because that basically, it's a fast fermenter, it's a clean fermenter. That's why we use it. We like to use it for research, and I like to use it for research. It doesn't give you a lot of aromas, right? I want to see the treatment and nothing else. Temperature control at 25 degrees Celsius, and 7-day skin contact. We press, we inoculate for malolactic fermentation, and we basically stabilize and bottle them. So a very simple process, so that really we're trying to showcase the grapes and nothing else, right? We're not trying to showcase our winemaking skills at all, other than not trying to make a faulty wine so you can actually see the grape differences. So would all of you agree that you can clearly see the difference between the negative and the positive wines, right? It's there. And obviously the extended maceration and the maceration enzymes in this instance did not have a huge impact in helping these wines look more than like the negative. But I personally do think that the longer hang time positive wine was slightly better than the one that was harvested at the same time as the negatives. Even though the sugar didn't move, it had riper characters, at least on the nose, and a little bit more weight on the palate. I think it's a little bit better to work with than the other alternative. Any other comments? Yeah, okay, George, ask me a question. An easy question.
I know. I'm not winemakers. I'm not an enologist. I'm not just 100% a viticulturist. And I'm from Fresno. So this hang time make me think, we make a lot of raisin in Fresno. Is that possible to cut the canes and make a semi-raisin type of grapes to make the wine?
We Where's Con? We actually tried trunk girdling. It didn't work that well. You know, you're going to get a lot of dried fruit character if you do that, by the way. We didn't really see, we tried trunk girdling to get the same kind of idea, and we didn't really see that it helped sugar accumulation at all, very much. So, not sure. You know, the thing is, you know, for commercial harvest, it's anyway closer to, well, you know, it's variable now because of a lot of other climatic influences and pressures. But if you're anyway harvesting at 27 bricks, you sort of have that longer hang time sort of worked in. But it depends on how different that red blotch fruit is. I have growers that tell me that they can get it riper. It can get to 25, the red blotch positive fruit, and I have others that tell me, no, they can never get it that ripe. But even if you can't get it as ripe, that little bit of extra hang time can help the extractability and help at least the phenolic differences that you observe. We're continuing to look at this. We really want to see an idea of this whole idea of years since infection, really figuring out the seasonal impact. And try and, the whole idea is really to give you some kind of measuring stick to evaluate what is the impact and help you make decisions. What do you do with these wines, right? Do you nurse them along or do you pull them out? Is there something you can do when you're stuck and you can't rogue? Is there something you can do to improve the quality of the wine you make from these vines? You know, when do you need to start really being worried about the presence of red blotch? What can you do? What can you measure? And at this stage, there seems to be a correlation between primary metabolite impact, something like just measuring bricks, and the overall impact that you're seeing on the wine. So that's something easy to measure. We've seen 100% relation between symptomology and virus status, but we haven't always seen 100% correlation between how badly impacted the vine looks in a specific year and wine impact. That we haven't seen a direct correlation. There's been years that the vine barely showed a lot of symptoms, and we saw more impact in years than it showed symptoms really early on. So we're still trying to figure that out. We really need a lot of years, a lot of seasons in the same site to be able to figure these things out, because otherwise, you know, among vineyard variability and a lot of other environmental influences, it can convolute the data, that it's difficult to make correlations. So it's slow, but we're sort of working away at this. And, you know, and the idea is by end of next year, our first, this SRI grant is going to finish. And we hope by the end of next year, we'll be able to pull all this research together. Not give you all the solutions, but at least be able to tell you, okay, this is what we know so far, and develop like a 2-page document for you to, this is what you can do. To the best of our knowledge, with everything we know so far, research-wise, this is the parameters you can use to evaluate and make decisions, and this is the options that you have. And we probably, you know, after that grant, we need to do a follow-up grant to investigate further, because the more you investigate, the more questions you often create, and you need to try and answer those questions. Research, unfortunately, is always much slower than we need it to be, but we will get there. As we have a lot of answers for leaf roll, we'll get those answers for red blotch as well. It's just a little bit trickier, as the entomologist told you this morning. Okay, so I'm the last person, so you're all allowed to like run away and escape. But I think Con, I'm not the moderator, I'm just the speaker. Anybody has a last question for me? Last question for me. If you think about something on the way home, just send me a message and I will answer you later. Well, thank you for your patience and thank you for your time.
Okay, well, thank you all for sticking here with us till the very end. Hope it was a good program for everyone in attendance and also on our live stream. And our next program is April 8th, correct? we're going to talk about smoke taint and all the other fun stuff. So thank you for coming. Thank you to all the speakers. Thank you for everyone that helped put the show together. Karen, Caroline, Justin, Maria, Lauren, Andrew, Chris. And we'll see you at the next event, hopefully. Thanks.